Hello, advanced biology students. This is our lecture 3 for chapter 12. We're going to focus on protein synthesis in this last lecture. We end it with looking at the genetic code, which is the unit that we use in molecular biology that consists of codons with unique arrangements that will code for each of the 20 amino acids that are found in proteins. Some of those amino acids are coded for by more than one codon. So when we think of the codon, we know that the messenger RNA transcript is going to be read in triplet code, or 3 nucleotides at a time. That would be the codon. They function as letters of the genetic alphabet. Genetic alphabet has only 4 letters that you know of: U, A, C, and G. The codons in the genetic code are all 3 base symbols long. Basically, permutations. If you do a permutation, there are 64 possible arrangements of 4 symbols, taking 3 at a time. They are often referred to as triplets. So genetic language only really has 64 words. Here you can see the genetic code, and you could use that genetic code, which we have done so in class. Finding the first base, second base, and then the third base at the end there would code for a particular amino acid. Remember that all of this entire process will begin with AUG. AUG right down in here, that is our start codon, or methionine is the amino acid. So all polypeptides have that methionine amino acid to start with. And then that process will continue. The process of translation will continue until one of 3 stop codons come into play. And the stop codons are UAA, UGA, or UAG. So here are the properties of the genetic code. And we didn't discuss these in detail in advanced biology in 10th grade. But you should have a basic understanding of why does this genetic code work. And the genetic code works because, number one, it is universal. With few exceptions, all organisms use the genetic code the same way, and they code for the same 20 amino acids with the same 64 triplets. It's also degenerate or redundant. There are 64 codons available for 20 amino acids. Most amino acids are encoded by 2 or more codons. It's unambiguous. Basically, the codons are exclusive. None of the codons code for 2 or more amino acids, and each codon specifies only one of the 20 amino acids. And it also contains a start and stop signals, punctuation codons, like the capital letter we use to signify the beginning of the sentence and the period to signify the end of the sentence. So the first step is transcription. Transcription in the process of protein synthesis is taking a segment of DNA to serve as a template for the production of a messenger RNA molecule. The genes unzip and expose the unpaired nucleotide bases. It's going to serve as a template for messenger RNA formation, and it's going to lose RNA nucleotides. It's going to have loose RNA nucleotides bind to exposed DNA bases using the rules for complementary base pairing, where C's will pair up with G's and A's will pair up with U's. Remember that in RNA we do not have the nucleotide thymine. It is replaced by a base called uracil. When the entire gene is transcribed into messenger RNA, the result is a pre-mRNA transcript of the gene. The base sequence in the pre-mRNA is complementary to the base sequence of the DNA. So we need to tighten up, or I guess sprucing up, that messenger RNA transcript before it leaves the nucleus. So transcription, looking at it in itself, is a single chromosome, consists of one very long molecule encoding hundreds or thousands of genes. The genetic information in a gene describes the amino acid sequence of a protein. So the information is in the base sequence of one side, the sense strand of the DNA molecule. The gene is a functional equivalent of a sentence. So genes are those functional units of the DNA. The segment of DNA corresponding to a gene is unzipped to expose the bases of the sense strand. That's the strand serving as the template. And ultimately, the genetic information in the gene is going to be transcribed or rewritten into a messenger RNA molecule. The exposed bases in the DNA determine the sequence in which the RNA bases will be connected together. And the enzyme involved in that process is called RNA polymerase. Pretty similar to the role that we saw in DNA replication, and we had the enzyme there called DNA polymerase, except now we're bringing in RNA molecules, so it's RNA polymerase. And the RNA polymerase connects the loose RNA nucleotides together. The completed transcript contains the information from the gene, but in a mirror image or a complementary form. So here you can see transcription taking place. Here you have the template strand, there you have the gene strand, and basically where this all began. Down here's your region called the promoter. So that's where the gene starts. And up here we have the terminator. So that's where it's going to stop. So transcription, the pre-messenger RNA is modified or processed before leaving the eukaryotic nucleus. Modifications to the ends of the primary transcript include a cap on the 5' end. So they call it the 5' cap. The cap is a modified guanine nucleotide. And essentially the cap itself is going to help a ribosome determine where to attach when translation will begin. And then you have the poly A tail. The poly A tail is going to consist of 150 to 200 adenines on the 3' end of the transcript. And it's going to facilitate the transport of messenger RNA out the nucleus, and it also inhibits degradation of messenger RNA by hydrolytic enzymes. So those are the roles of the caps. And again, that is new information for AP biology. In transcription, the pre-messenger RNA is composed of nucleotides called exons and introns. Exons are expressed, introns are not expressed. Often we remember exons expressed, introns are in the way. The introns occur in between the exons. The exons basically are going to be the nucleotides that are going to carry the message for the translation into a protein. So it allows the cell to pick and choose which exons go into a particular messenger RNA. RNA splicing, the primary transcript consists of some segments that will not be expressed, introns, and the segments that will be expressed, exons. So in the higher eukaryotes, the removal is done by spliceosomes, and basically spliceosomes that contain these small nuclear RNAs, or you call them snRNAs. The small nuclear RNAs serving as spliceosomes are going to use a ribozyme, an enzyme made of RNA rather than just a protein, to cut and remove those introns out of the way. And the remaining exons are then spliced back together. And then ultimately the result to that is a messenger RNA transcript. In prokaryotes, however, introns are removed by self-splicing. That is, the intron itself has the capability of enzymatically splicing itself out of pre-messenger RNA. So if we look at the diagram, here you can see the DNA. We see the exons and introns. We see the pre-messenger RNA, 5' to 3', exons and introns. There's your cap, 5' cap, 3' poly tail. And now you have the spliceosomes coming in, and it's going to remove those introns. And ultimately, you're going to bring that messenger RNA mature transcript together by pulling those exons, fusing them together. And ultimately, then you could have that leave the nuclear region and go into the cytoplasm where it will meet up with the ribosome. Transcription itself, what are the functions of introns? Now, this is a lot of new information is coming out about the role of introns. Scientists at one point, when I was sitting in your seat or walking in your shoes as a biology student, we thought it was just junk DNA. There was no use for introns. But as research continues in molecular biology, we are finding that indeed they do have an important role in organisms. So functions of introns. As organismal complexity increases, the number of protein coding genes does not keep pace. So the proportion of the genome that is introns increases. So here are some suggested possible functions of introns. Exons might combine in various combinations. This would allow for different messenger RNAs to result from one segment of DNA, and ultimately the introns might regulate gene expression. So introns may encourage crossing over during meiosis. They will also keep certain exons in and maybe take some out. So an exciting new picture of the genome is slowly emerging as we continue to seek answers in this particular field of biology. So moving genes between species, there is a really interesting, I guess, side note in your book that I would read over. It's on the green fluorescent protein, or GFP, and we might be able to do the pGlow gene lab in class. But ultimately, the green fluorescent protein is a bioluminescent, and one of my favorite things about organisms within the ocean is their property or characteristic of bioluminescence. And we do see this often in jellyfish. The Eichorea victoria jellyfish is normally transparent, but when disturbed, it releases this protein which fluoresces it with a green color. Now, you've heard of this word before. Think of fluorescence when we looked at photosynthesis, and we talked about the cyclic pathway of photosynthesis, where when you fluoresce, you give off that heat and light energy. So scientists were able to isolate the fluorescent protein and develop a molecular tag. The GFP gene is inserted after the promoter region of another gene in a different organism. And ultimately, RNA polymerase binds to the promoter and will initiate transcription of the gene, which then can be expressed and the GFP protein be made. Expression of the GFP gene produces a glow in organisms besides the jellyfish. So we have inserted this in. We use some of this as molecular markers. We have put this in certain plants, and ultimately what we see is that you could cause these animals to glow. Also, this is done a lot in mice. When I taught up in Maine, there was a very well-known worldwide genetic research lab called the Jackson Laboratory. And in there, they did a lot of stuff like this. They had glowing mice in their labs. Also, part of medical biology, they would have parts growing on mice, like ears and stuff. So, and we'll look at that a little bit later in the next chapter, what biotechnology can do. So these molecular tags can reveal cellular locations and distribution of proteins within a cell. And here you can see the result. There's the jellyfish. These are the glowfish that you often could buy in a particular store, fish store. And then here you can see us. Ultimately, here in this picture, you are looking at the genes, or not genes, cells that have the gene in it, the GFP gene. So the second step of protein synthesis is translation. Translation is the sequence of codons that are going to be read in the messenger RNA at a ribosome that's going to determine the sequence of amino acids into a polypeptide. A nucleic acid sequence is translated into a protein sequence. Ultimately, transfer RNA or tRNA molecules have two binding sites. One associates with the messenger RNA transcript. The other associates with a specific amino acid. So each of the 20 amino acids in proteins associates with one or more of 64 types of tRNA. Francis Crick, in his research, came up with this wobble hypothesis. And the wobble hypothesis predicts that the third position in a tRNA molecule, or the anticodon, doesn't obey the A, U, or GC configuration rule and can be variable. Ultimately, translation then of an mRNA transcript associates with the rRNA of a ribosome in the cytoplasm, or a ribosome associated with the rough endoplasmic reticulum. The ribosome is going to read the information of the transcript, and the ribosome will direct various types of tRNA to bring in the specific amino acids. The tRNA specifies is determined by the codon being translated in the messenger RNA. And that's where you use that genetic code. tRNA molecules come in 64 different kinds, all very similar, except that one and bears a specific triplet on the 64 possible called the anticodon, and the other end binds a specific amino acid type. tRNA synthetase attaches the correct amino acid to the correct tRNA molecule. All tRNA molecules with specific anticodons will always bind with the same amino acid. So here you can see it. Here's where you have the amino acid attached. Down here you have the tRNA anticodon. And the tRNA, given its name because it looks like a T, these are all little hairpin loops here. So at the one end you have the anticodon, and up here you have the amino acid attachment point. And then these little bands are held together just like in the DNA molecule. You have those weak forces of hydrogen bonds. So there you can see the messenger RNA. Here's its codon. Remember, it's the codon that you use in the genetic code. It's not the anticodon. So if you were to look up CUU in the genetic code, that would code for the amino acid leucine. So ribosomal RNA is produced from DNA template in the nucleus, in the nucleolus of a nucleus. Remember, that's where ribosomes are made, and packaged with proteins into large and small ribosomal subunits. The completed ribosome has 3 binding sites to facilitate the pairing between tRNA and messenger RNA. Those 3 sites are abbreviated EPA for exit, peptide, and amino acid. So the large ribosomal subunit has enzyme activity that creates a polypeptide bond between those adjacent amino acids. The polypeptide bond is created many times to produce a polypeptide which folds into its 3-dimensional shape and ultimately will help become a protein. Remember the 3-dimensional shape, that's examining those levels of protein structure: primary, secondary, tertiary, quaternary. So here you can see a diagram of that coming in. Here you have the amino acid site. So this is where the amino acid would come in, and the anticodon of the tRNA would ultimately come in with the codon of the messenger RNA. Here's where the polypeptide, in the P site, the polypeptide would build from. So the polypeptide chain would come out of there, and then the E site's where it would exit. So the tRNA would exit there. It occurs in 3 steps: initiation, elongation, and termination. So the first step, initiation. Components necessary for initiation are a small ribosomal subunit, messenger RNA transcript, initiator tRNA, and large ribosomal subunit, and initiation factors, special proteins that bring the above all together. The initiator for tRNA always has the UAC anticodon, because remember the messenger RNA is AUG, so it always carries the amino acid methionine, capable of binding to the P site. A small ribosomal subunit attaches to the messenger RNA transcript. The beginning of transcript always has that start codon AUG, and initiator tRNA UAC attaches to the P site, and a large ribosomal subunit joins the small subunits all together. So here we have initiation taking place. And this is diagrammed in your book. Elongation refers to the growth in length of the polypeptide. The RNA molecules bring their amino acid pairs to the ribosomes. Ribosomes are going to read a codon in the messenger RNA. It allows only one type of tRNA to bring its amino acid. It must have the anticodon complementary to the messenger RNA codon being read, and the incoming tRNA joins the ribosome at its A site. So methionine of the initiator tRNA is connected to the amino acid of the second tRNA by the peptide bond. The second tRNA moves to the P site, that's translocation, and the spent initiator tRNA moves to the E site and exits. The ribosome reads the next codon of messenger RNA, and this process would continue and continue, and we'd see the building of that polypeptide. So it allows only one type of tRNA to bring in its amino acid. It must have the anticodon, complement the codon, joins the ribosome at its A site. The dipeptide on the second amino acid is connected to the amino acid of the third tRNA by that peptide bond. So you can see how they continue to build and are held together via peptide bonds. So here's our elongated polypeptide chain that is forming. And the process would just continue and continue and continue as the ribosome continues to read that messenger RNA transcript until one of three stop codons comes into play. So that is elongation. And ultimately then it would end with termination. So previous tRNA moves to the P site, spent tRNA moves to the E site and exit. And the ribosome reads that stop codon at the end of the messenger RNA. UAA, UAG, UGA are the three stop codons. It does not code for an amino acid. But basically what happens is a protein called a release factor is going to bind to the stop codon and cleaves the polypeptide from the last transfer RNA molecule. The ribosomes releases the messenger RNA and disassociates into subunits again. And the same messenger RNA may be read by another ribosome to continue to produce that particular protein. So here you can see termination taking place. So a summary of protein synthesis in eukaryotes is as such. You could read over these steps. They are also diagrammed in your textbook. This is what we just spoke about. It's a little bit more detailed than what we learned in advanced biology, but not too much. So ultimately, gene expression then. A gene has been expressed when its product, a protein, or an RNA is made in and operating in the cell. Translation occurs at ribosomes, and some ribosomes are free in the cytoplasm, while others are attached to the endoplasmic reticulum, ultimately giving it the rough appearance, or rough ER. The first few amino acids of a polypeptide act as a signal as a signal peptide with information where the polypeptide belongs. The polypeptides can either belong in the cell or be secreted from the cell itself. So when we think of those proteins, they could become part of a cellular process inside, or maybe something that is needed external to the cell. So we'd have to get rid of it out of the cell via exocytosis. Some polypeptides are processed further by the addition of sugar, phosphates, or lipids in the rough endoplasmic reticulum. Ultimately, transport vesicles will carry the proteins between organelles and the plasma membrane as appropriate. So we have those transport vesicles, and before some of those proteins would be used, they may go to the Golgi apparatus where they're modified even more, hence getting some of those other additional components, and then shipped out via transport vesicle. This all makes sense when we did cell structure function. You can see how all relates now when we look at that endomembrane system. So that's it, everybody. That finishes chapter 12, molecular biology of a gene. Our test will be coming up, so please study. And if you have any questions, jot them down, and we'll review this a little bit in class and go over the chapter 12 student focus questions. Have a great day.